Hello, everyone. I'm talking from a very sunny afternoon here in Cambridge. This is the second time that Cambridge, UK is hosting at the RNA Collaborative Club. Very exciting to be host again. There are a number of you just coming in still. So I'll wait maybe a minute or so until the numbers stabilize. But welcome. Hope you've got a cup of tea or something ready. Not that you would need a caffeine in addition, I don't think, but we'll have two great speakers for you. Right, numbers are still going up. Very exciting. Okay, if you're just joining now, welcome to the RNA Collaborative Club. This time it's Cambridge, England's term. And we've got two speakers here from Cambridge where it's sunny, beautiful, and hopefully it will stay like this for a couple more days. Anyway, right, instead of just talking and filling the air with nonsense, let me start by introducing the first of our two speakers, who is Kathryn Lilley, who's a professor here in the Department of Biochemistry at the University of Cambridge. And she has a strong track record in developing new tools to ask exciting questions inside the cell. Her major technology has been mass spectrometry. And you might have guessed that she spent a lot of time and is spending still considerable time looking at proteins. But more recently, she's been very interested at the interface of RNA and proteins, and not only how these two types of molecules interact inside the cell, but where inside the cell they interact. And I'm very excited to present her to you. And the title of her talk will be The Spatial Organization of the Cell. And before I hand over to Kathryn, just to give you the heads up, please use the Q&A instead of the chat for asking questions. And think about the questions during the talk, but please wait till the talk is done, and then we'll go together into the Q&A. And with this, I hand over to Kathryn.
Thank you very much, Eric. I've just hidden all my floating panels, so can you hear me?
We can, loud and clear.
Excellent, right, wonderful. So thank you very much for the invitation to speak to you today. As Eric alluded to, my lab has a history of technology development, and what I want to do today is to talk about largely two methods that we have developed, both centered on determining the spatial organization of the cell. One giving maps of the subcellular proteome, and a more recent one that gives a map of the subcellular transcriptome. So I will start off by really making the beginning of this talk rather protein-centric, but I hope by the end of it you'll realize why I've done this. So proteins have to traffic to the right part of the cell. Cells are not just bags of protein, they've all got to get to the right place where they can carry out their function. And how they move and where they move to is dictated by many different features of proteins: sequence motifs, post-transcriptional modification, post-translational modification, who they're interacting with, their structures, and even localized translation. And there's more and more mounting evidence that when something goes wrong, proteins end up in the wrong part of the cell, and this gives rise to various diseases. And what can go wrong can be in any of the different features that I've just described, from sequence motifs to aberrant post-translational modification. But of course, RNA also has to get to the right part of the cell to carry out its function. Many different types of RNA molecule, as you all know, and they have to be trafficked to the right part of the cell to carry out their function. And there's evidence, again, that when this goes wrong, this can give rise to a variety of different disease states. So it's important for us to know where proteins and RNA are located in cells. And there are two different ways in which we can think about this. we can think about static maps of the subcellular location of either the transcriptome or the proteome. And by knowing where molecules are, this is going to give us an insight possibly into their function. And by knowing which molecules are present in certain subcellular niches, this is going to give us an idea of the functions that can take part in this particular subcellular niche. But we can also look at these maps in terms of moving maps, so looking at them dynamically. So if we find out where a protein or an RNA molecule moves to in response to a perturbation, this may also give insight into its function. And again, aberrant localization may shed light on some disease mechanisms. And I would argue, and hopefully you will agree with me by the end of this talk, that it's very nice to have overlapping maps of both. So imagine that we can get a map of as many proteins as we can measure and as many transcripts as we can measure, and have these nice overlapping maps from which we can see how RNA and protein is orchestrated together. So how it exists, how RNA and protein exist together. And this will give us an idea of localized translation, because we might find that a messenger RNA and the protein which it encodes are at the same part of the cell. It may also give us some insights into cellular processes which involve orchestration and movements of both RNA and protein, and also may help us understand the disconnect between the transcriptome and the proteome. And this has been well documented and well argued over many years, why if you look at the quantities of transcripts and their cognate proteins, that they don't always match. So may give us some insights into why there is sometimes this disconnect between the two. So my lab were very busy during the lockdown writing reviews. So I won't labour the point, but we've written a couple of reviews which talk about the methods that are available to look at, firstly, the subcellular proteome and also the subcellular transcriptome. So for the proteome, the method that you use will determine or will be determined by what your question is. Whether you're interested in knowing where a particular protein is, in which case you will use some sort of immune fluorescence, whether you want to know what proteins are present in a particular compartment, in which case you could use proximity tagging or some sort of organelle enrichment, or by cell, if you want a cell-wide map, in which case you might consider using protein correlation profiling methods, which I'll talk about in a moment. Now, all these methods are beautifully paralleled in the RNA world, again by RNA species, where you would use fluorescence in situ hybridization and other similar methods. By compartment, again, you could use apex-seq and use proximity tagging methods. You could try and enrich a particular subcellular niche and determine the RNA present in that niche. And also, arguably, we could also use correlation profiling methods for RNA too. So I point you to these two reviews that have been driven by a very talented PhD student in my lab, Josie Christopher, and were both published last year and the beginning of this year. So I want to talk now about correlation profiling methods, because this is what the methods which I'm going to talk about, both the protein method and the RNA method that we've developed in my lab, are based around. So protein correlation profiling is not new. It was first described in some detail by Belgian scientists Christian de Duve and Albert Claude, who received the Nobel Prize for having discovered the lysosome and the peroxisome. And they argued that you don't need to purify any subcellular niche to homogeneity to work out where proteins are in cells. In fact, they argued that it's impossible to be able to purify certain subcellular niches away from one another because their physical properties are too similar from one another. So they argued that if you carried out some sort of subcellular fractionation, then proteins that belong to the same place as one another will have the same abundance profiles through these fractions. So for example, this is a very stylized example of this. Let's imagine we've carried out some sort of fractionation, and in our first fraction, we've got lots of lysosomes and a bit of Golgi apparatus. In our second fraction, we've got more Golgi and fewer lysosomes, and so on and so forth until our final fraction, which is more nuclear. And no fraction is pure, but if you look at marker proteins from those particular organelles, then lysosomal proteins should look a bit like this. Golgi proteins should look a little bit like this. ER proteins like this. mitochondria, and nucleus. So no fraction is pure, but if you look at the correlation profile of marker proteins, they will all have the same correlation profiles as each other. And then when you measure the correlation profile of something which is unknown, you simply match it. So this one would be a novel Golgi protein. Now, in Deduve and Claude's day, they had to develop methods to work out what these correlation profiles were. And the only way they could really do that was to use enzyme assays. But of course, these days, we can use quantitative mass spectrometry to be able to do this. And later on in my talk, next generation sequencing, if we're actually looking at RNA species present in these subcellular fractions. But let's just concentrate on proteins at the moment. So I'm going to talk to you about the methods that we've developed. Our protein correlation profiling method we've been working on for almost 20 years now, and we call this Loppet, the localization of proteins using isotope tagging. And it's a method that gives us a map of thousands of proteins in a single experiment. We also more recently have developed the RNA equivalent of this method, which we call Lorna, or the localization of RNA. And these are supported, particularly the protein methods, by a series of R packages, which are submitted to Bioconductor, which allow us to classify proteins to different subcellular locations, and also to be able to give probabilities of whether those proteins are moving if we're looking at a dynamic system. We also have a method that sort of ties the two, the Loppet and Lorna together, which I'm not going to go into detail, which is a method to very efficiently sample the RNA binding proteome, which we call OOPS. But Loppet first. So what we do in our Loppet protocol is to crack open our cells. We do this using a ball-bearing homogenizer, and the idea is to be as gentle as possible to leave the organelles intact. We have then two different versions of subcellular fractionation, one using differential centrifugation, which we call Loppet DC, and one using equilibrium density centrifugation, which we call hyperloppet. And in our hands, the matrix that we use for carrying out equilibrium density centrifugation is iodixanol, which is a self-generating gradient matrix. So the gradient is formed upon centrifugation. So whichever way, whether we're using differential centrifugation or equilibrium density centrifugation, we end up with a series of fractions, which we then use quantitative mass spectrometry methods. We use isobaric tagging. I won't go into the details of this, it's in all our papers. And basically, we end up with a series of correlation profiles, which we can then use fairly sophisticated machine learning tools to be able to classify proteins to different subcellular niches. The type of results that we would get is something that looks a bit like this. This is a principal component analysis, which is separating out our data in various principal components. I'm showing the first two here. Every spot is a protein. If it's colored in, it's either a marker protein to a particular organelle, or it's been classified to that organelle using a machine learning tool. And the way to interpret this is we've actually got sub-nuclear fractionation in this particular approach that we took. This was using mouse embryonic stem cells and the hyperloppet, the density gradient method. So anything in pink is associated with chromatin, anything in pale green is non-chromatin associated nuclear protein. On the top left-hand side, we have the secretory pathway all the way from the endoplasmic reticulum through to the plasma membrane. We have the cytosol in blue down here, the mitochondria and peroxisome in the top right-hand corner. And we also get clusters of proteins that are associated with large protein complexes. In brown, we have the proteasome, and in the center of the plot, the two ribosomal subunits. What you will see is a series of gray spots. And what these represent are proteins that are in multiple localizations. So they're not classifying to a single localization, but using our machine learning tools, we can recapitulate the correlation profiles and determine which different subcellular niches those proteins belong to. We can now identify and classify and therefore give an address to 8,000 proteins per experiment. In the stem cell example from a few years ago now, although there was a significant amount of go-ontology associated with the subcellular niches that the proteins were thought to belong to, very few of them had been empirically derived. And it's important to say that around 50% of proteins have mixed localization. So we can use our Loppet method to produce static maps or dynamic maps. So static map, it's nice to have them. You can use them to assign proteins to specific localization, different locations. You could use them to find, if you use semi-supervised analysis methods to find clusters of proteins that may represent new organelles, or even find components of poorly characterized subcellular niches. And over the years, we've used it in a variety of different ways, of a very broad range of different biological systems. For example, we've mapped membrane proteins to the thylakoid membrane and plasma membrane in Silica cystis. So this wasn't very clearly known before we applied Loppet to it. In a very beautiful study with my colleague in the biochemistry department here, Ross Waller, what we've done is to map proteins to 26 different subcellular niches in the obligate parasite Toxoplasma gondii, including being able to map for the first time proteins to some of the very specialized organelles that this parasite has, to be able to glide across the surface of host cells and infect them. And in a study that we carried out some years ago now with the Human Protein Atlas group in Stockholm, we compared and contrasted hyperloppet with a mammalian cell line with high throughput immune fluorescence. So using a very orthogonal method to try and compare and contrast whether we see the same sorts of localizations. And what we determined by both different methods was that there was a very healthy overlap, and also in their hands, also 50% of proteins are residing in multiple locations. So the messages from this, proteins, many of them have got mixed localization. What we're doing is not a single cell method, so we have to be mindful of this, and it results of mixtures of cells and cell states. And also, if you have proteins in different locations, you don't know whether those proteins are interchanging pools or discrete pools of proteins. We can also produce dynamic maps. And here we've got more of an informatic nightmare, and I shan't go into details, but we need to be able to not only classify proteins to particular places, but we need to know between two situations if those proteins are moving. So we need two sets of probabilities. Is a protein residing in a particular place, and does it move? And also we need to be able to be mindful of the fact that proteins in mixed localization may be moving to a single localization or even different mixed localizations. And we have a series of packages, including a recent one, which you can read about on bioRxiv, which is a Bayesian approach to be able to work out which proteins with confidence are moving from one place to another. We have applied this dynamic method to look at all sorts of different systems. One was published last year, which is to look at how proteins rearrange themselves during the stages of the innate immune response by eliciting the innate immune response in a monocytic cell line using a lipopolysaccharide. This is in collaboration with Andrew Smith and carried out by two very talented postdocs in my lab, Claire Mulvey, who has since left the lab, and Lisa Breckles. So the idea here was to capture the spatial proteome of this system and how proteins move in response to the activation of a pro-inflammatory phenotype, but also a more fundamental question, which is when you do something to a cell, do the proteins relocate or do they change in abundance, or both? So we were able, by using a normal quantitative proteomics on a total cell lysate, and also Loppet using the hyperloppet method, to disentangle that question. And what we found was that many proteins are moving, including the set of Rho GTPases, which is cytosolic in untreated cells, but in response to treatment with LPS, they move lock, stock and barrel to the plasma membrane, but in all cases with no change in their protein abundance. So this is quite an important message, because a lot of proteomics, in fact, all omics studies, tend to ignore the spatial part of it. So these movements wouldn't have been highlighted if we just looked at total cell lysates of proteins and looked at changes in protein abundance. And if proteins are moving from A to B, it's difficult to know if they're shuttling or if there's degradation in one location and synthesis at another. Okay, so we can do this for proteins. We've been doing this for proteins for some while now, and we've published many papers using Loppet, which shows that it works. So the question was, can we use the same fractionation methods and do the same for RNA? Can we carry out RNA correlation profiling? Well, yes, we can. Our method we've called Lorna. It's submitted to bioRxiv, and at the bottom of the slide, it'll tell you how to find that paper. And what Lorna is a method which allows the simultaneous mapping of the transcriptome to subcellular organelles, membraneless organelles, and cytosol in a single experiment. So this has been a labour of love over the last 5 years or more in a collaboration with Ann Willis's group, and was funded by the Wellcome Trust. And here we are somewhere or other celebrating something which I can't remember, but we all look very happy, you have to admit. So this has been more of a journey than I think we imagined it would, because the methods that you need to use to fractionate the cells need to be very carefully thought out. Now, we're not unique in trying this, other people have tried it too, but methods can be fraught with RNA fractionating based on its size rather than which organelle it's associated with. And it took us a while to come up with methods that were able to fractionate organelles and the RNA inside them and not the RNA based just on its size, and also that we're mindful of keeping RNA intact and nice and healthy during the process. So Aneko Villanueva and Mo Elzek in the lab tried very hard and eventually got very nice methods, both using density centrifugation, so this is D-Lorna, and sedimentation methods, which is S-Lorna, to be able to fractionate cells in such a way that we can measure, using next generation sequencing, RNA correlation profiles. So this is what we get. So the picture is not quite as fulsome as the protein equivalent, and that is because the majority of RNA species are in fewer locations. But we're able to use marker RNA molecules and see clusters which are basically the cytosol. We've got two nuclear clusters, the nucleolus and the nucleus itself, the endoplasmic reticulum, and also the mitochondria. And there are a lot of RNA species which are in multiple places. So we can compare the profiles, the correlation profiles we have for marker RNA species with those of unknown species, and then able to classify RNA to the different subcellular niches. We also found an interesting cluster, which for the moment I'm going to call cytosol light. So these are happy U2OS cells, nothing wrong with them, but we have this sort of separate cluster, which we're calling cytosol light. And all the informatics work was a gargantuan task. The majority of it was done by Tom Smith. So if we compact the nucleus fractions into the nucleus and the ER in the mitochondria, because there's not a huge amount of mitochondrial RNA, into membrane, we end up with these very nice ternary plots. And we can see that the majority of RNA is cytosolic. Also, because of the experimental design with spike-ins, we can work out the absolute proportions of RNA species that are shared between multiple different places. With any new method, you want to test it out. So Vicky Pitzinger in Ann Willis's lab carried out various fish experiments to then validate what we were seeing. So, for example, Mallet1 is a long non-coding RNA, which is nuclear, and it's down towards the nuclear end of our triangles. Calpain2, the messenger RNA, is cytosolic, which we think it's cytosolic too, at least Lorna does. And finally, PIG-T, which is a messenger RNA coding for an ER protein, is towards the membrane apex. So it makes some sort of sense that we're seeing. We can also then have a look at the features of RNA molecules, which we're putting in different parts of the cell. RNA species, messenger RNA that are destined to be translated into proteins that are going to go through the secretory pathway, they are on the axis between the ER and the cytosol. And interestingly, I haven't shown the data here, the position of the first transmembrane spanning region of the RNA transcript correlates with where the RNA is along this axis. We can also find at the sort of ER apex RNA species that we wouldn't anticipate being there. When we look at the Go terms for these species, we see that they're overrepresented in membranes, cell adhesion, and cytoskeletons. So these themselves are not membrane proteins, but are coding for membrane-associated proteins and proteins which are involved in the cytoskeleton. And Vicky did a very nice job to test this out for a couple of them to actually validate using fish. So it's all making some sort of sense. And so because we're confident in it, we can then look at features which are driving the RNA species into different places, and also the distribution of different types of RNA. So for example, if we look at long non-coding RNA, we were somewhat surprised to find that more than we imagined was cytoplasmic. And we can have a look to see, or rather cytosolic. We can have a look to see the features of the long non-coding RNA species, which are really discriminatory between those that are nuclear and those that are cytosolic. And what we find is that the majority of the features which are giving this discrimination are to do with size, AU content, and if the long non-coding RNA itself is polyadenylated. Okay, so we've now invented Lorna. We've also got Loppet. Can we not join the two of them together in a single experiment? So to do this, we decided on a dynamic system to test out, which is the unfolded protein response. So what we did was to treat our cells with papsigargin that blocks the calcium, one of the calcium transporters in and out of the ER. You end up with an increase in unfolded proteins, which ultimately ends up in phosphorylation of EIF2 alpha and cessation of translation. It's inhibited at the ER, and the transcripts go off into stress granules. So this is what we knew. So how can we combine Loppet and Lorna together? So we carry out, in this case, density centrifugation. And then for each fraction, what we do is to carry out phase separation. So for the aqueous, this is where our RNA is going to be, and that gets sent off for next generation sequencing. And the organic is where the protein is going to be, and so that gets sent off for quantitative mass spectrometry. So the aqueous is Lorna and the organic is Loppet. So one is going to give us the map of the transcriptome and one is going to give us the map of the proteome. And because they're from the very same fractions, we then hopefully can overlay them. So this is what we get. The top are the RNA plots. On the left-hand side, we have the control, and on the right-hand side with papsigargin, so having elicited UPR. The bottom are the Loppet plots. So this is the protein, again, on the left-hand side, the control, and on the right-hand side with papsigargin. And I'm just going to talk you through now our results. I've highlighted this is a high-level summary. At the bottom, a movement of proteins from the ribosome into what I'm going to convince you, I hope, is cytosol light. And up here, the movement wholesale of ER-related transcripts into cytosol light, and a persistence at the ER of some transcripts, which I'll talk about in a moment. So proteins first. There aren't that many high confidence movers in our system, so the proteome is somewhat bulletproof in UPR. What we do see is a fair few Golgi proteins which are moving around, but of interest, 28 proteins which were classified as ribosomal in our control that move to what we're calling cytosol light. And these proteins, many of them are associated with stress granules and P-bodies, including STAU2 and the C4 NOT complex and PAPC1. And what we think is happening, in fact, what we've shown is happening, is that these proteins are moving sideways into fraction 6. And fraction 6 is where we have got an enrichment of granules. So we know we're forming stress granules by treatment with papsigargin. We can actually have a look at fraction 6 under the microscope. in a system where we have fluorescently tagged G3PB1. And so this would suggest that cytosol light is likely to be stress granules. We can then look at the correlation profiles for the transcripts, and we've got, again, fraction 6 is cytosol light, and start to have a look what sorts of transcripts are moving into cytosol light. And essentially what we're seeing is transcripts that were at the ER apex moving into cytosol light. And Vicky Pitzinger again has tagged a couple of these and shown that these transcripts rather are going into stress granules. So we can have a look now to see what the features are of these transcripts that are going into stress granules. And it's really very interesting. They're more likely to have a higher AU content. They're more likely to be longer. And interestingly, they are enriched or de-enriched for codons, which are decoded by tRNA molecules where the wobble position is post-transcriptionally modified by cuisine, so cuisinylated transcripts. So here relocalization seems to be dependent on size and AU content. These are messenger RNAs. So when we have a look at long non-coding RNAs with exactly the same physical properties, what we're seeing is that they are not going into these stress granules. So the bottom line is that the long non-coding RNA with the same properties don't relocate to granules upon UPR, irrespective of their AU content and their size. So just finally, I alluded to the fact that there are a set of transcripts that remain associated with the ER during this process, so they still cluster together. And when we had a look what these were and went into ENCODE data, they seem to associate with EIF3D. So this would suggest that they're going through potentially a different translation initiation pathway and are persistently translated during this process. When we knock down EIF3D, and this is work of Rob Harvey in Ann Willis's lab, what we find is that there is a reduction in cell migration, and the transcripts are coding for proteins that are enriched again in terms to do with the cytoskeleton and cell adhesion. So in summary, for Lorna and the unfolded protein response work that we've done, and as I say, this is all in this bioarchive paper that you can access, we've identified for the first time RNA in phase-separated granules at the same time as all the other RNAs of cellular location. So we've not tried to purify it. It's part of our correlation profiling has given us the contents of these phase-separated granules. We've shown that long non-coding RNA is predominantly located in the cytoplasm, that cytoskeletal RNAs are localized at the periphery of membranes consistent with localized translation. Membrane-associated RNAs are more readily relocalized to granules upon UPR. And the transcripts that are relocalized, this seems to be dependent on their AU content and their size, and the fact that there is something going on with cuisine-modified tRNA decoding. Long non-coding RNAs do not relocate to granules, even though they have very similar physical properties to the messenger RNAs that do. And we've shown that translation via a EIF3D dependence still seems to persist during UPR. Overall, I hope I've sold you on the idea that mapping subcellular location and how it changes gives insight into function, that half the proteome exists in one subcellular location, that perturbation may lead to relocalization of proteins with no change in their steady state abundance, that we can map the spatial transcriptome in the same way that we're doing for proteins and have been doing for a while, and that we can form different maps of both the transcripts and the proteins from one single experiment. And we hope that these methods will be taken up by the community and give essential insight into mislocalization and its source. So I've got a lot of people to thank, particularly Anne's group.
Kathryn, sorry, would you mind
facility and people that have funded this work. So thank you very much.
Oh, can everybody hear me? Sorry, we are quite over time, unfortunately. Very quickly, there are two Q&As. Is this method applicable to different cell types? in a tissue? Liver? Yes. Correct answer. I mean, fast answer. Can we use Lorna to also find out viral RNAs in different compartments of a cell?
Yes.
Sounds good. Brilliant. For more information, please contact Kathryn. And Kathryn, can I ask you to unshare your screen? I'm very sorry. I need to find an electronic way of waving my hand out. Alex, are you there?
Yes, I am.
Because, after this rich talk, I want to next introduce Alex Borodavka, who is a group leader. I mean, I was thinking, I mean, it's kind of weird. Let's say it's almost like an assistant professor, I guess, in the American system. Anyway, Alex does his own independent research, and he's particularly been excited about rotaviruses, and today he's going to tell us about molecular mechanisms underpinning RNA-controlled self-assembly of multi-segmented viral genomes. And off you go, Alex.
Thank you, Eric, and thanks for giving me the opportunity to present. And I think it's going to be quite difficult after overwhelmingly exciting talk from Kathryn, but I hope I can address some of the questions, because some of the questions just asked about viral RNA localization. So yes, as my title suggests, I'm a big fan of viral RNAs, but generally RNAs. And that sort of brought me into rotavirus field, because rotaviruses, and I hope you can see my screen now. This is a little bit unusual when you, given the webinar, you can't really see your audience. I hope
all good.
It's, so one of the implications of genome segmentation in quite a few RNA viruses, and this includes influenza A virus or rotaviruses or reoviruses, is that these viruses can co-infect the same cell, and then essentially due to genome segmentation, they can sort of produce this mix and match effect, or what we call reassortment in virology. And so that results in this so-called reassortant viral strains. And usually this manifests in the production of viruses with different antigenic properties. So in a way, it's a rapid way for these RNA viruses to evolve due to reassortment, and usually causes something what we call antigenic shift, as opposed to slow mutation accumulation causing antigenic drift. And so one of the reasons why I like rotaviruses is that in flu, for example, which have 8 RNA segments, these can go up to 11. So we actually have 11 RNA molecules all together packed in one particle. And so that obviously poses a lot of interesting conundrums with the virus, because structurally, these viruses somehow need to assemble each time stoichiometrically a new virus particle where you have exactly 11 distinct RNA molecules. Otherwise, the whole genome won't be infectious. And so if you do try and do this, work out this using bioinformatics or computationally, it becomes a very complex problem, because essentially just simply by looking at the energetics of RNA-RNA interactions, and this is just intermolecular RNA-RNA interactions. So here we're just looking at the entire genome of rotavirus in this diagram. And you can see the whole genome is about, depending on strain, 18.5 kilobase or 19 kilobase altogether, all 11 RNA segments shown here. And we're just looking at Watson-Crick interactions, stable interactions below 10 kilocalories per mole between 11 distinct RNAs. So obviously this is a very simplified model, and it doesn't take into account intramolecular base pairing, and it doesn't take into account translation of our RNAs, because each of these RNAs which will be packaged, they also somehow need to be spatially separated, and probably temporarily separated during virus infection, because they can both serve as substrates for genome packaging, but also for translation. And so Daniel Desireux here, who's a talented bioinformatician in my group, he's been developing several tools. I don't have time to talk about RNA-RNA interactions and using his recently developed tools for predicting RNA-RNA interactions and virus sequences, but please do feel free to get in touch and discuss this if you're interested in predicting RNA-RNA interactions. And so we're trying to reduce this complexity. We're trying to understand how viruses with segmented genomes evolve via these RNA-RNA interactions. And so a few years back, we discovered that there are a number of RNA binding proteins with sub-nanomolar, nanomolar affinity towards RNA, single-strand RNA, that bind to RNA very tightly, and they're always produced by viruses. And they're essentially what we discovered, they are RNA chaperones. They're usually multimeric RNA binding proteins. They're highly enriched during viral infection. And the way we discovered that these proteins possess RNA chaperone activity is by using single molecule techniques. So my group has always been a big fan of using single molecule fluorescence tools. One of the tools I've been particularly big fan of is called fluorescence cross-correlation spectroscopy, or FCCS. And so essentially what this technique allows us to do is we can fluorescently label distinct RNA molecules. In our case, we were interested in interactions between segment 11 and segment 10. We label them with spectrally different distinct dyes. And then we're looking at essentially formation of stable complexes and their co-diffusion, which manifests in this non-zero cross-correlation in this case. When RNAs are not interacting, we get zero cross-correlation essentially. And so what we discovered back in 2017 is that upon incubation, co-incubation of 2 RNAs, in this case, an addition of entire complex of 11 different RNAs, in the presence of RNA chaperone and sp2, when we saturate this complex, RNA complex, we get a stable cross-correlation function, which persists even when we remove an sp2 by proteinase K digest. So that sort of suggested that an sp2 does promote or stabilize RNA-RNA contacts. And so more recently, we started examining structure of an sp2, and the most exciting finding from recent studies came out from single molecule, combination of single molecule fluorescence studies and combination of cryo-electron microscopy. And so this story is basically about this C-terminal region, which we knew was absolutely critical for virus replication. If we remove this small 17 amino acid region of this RNA chaperone, the virus does not replicate. However, the protein itself still assembles in this beautiful optimeric structure, so donut-shaped structure, as you can see here. What was interesting about this region, and this is why we focus on this region, is in essence, this is completely pretty much unstructured region of an sp2. And so what we discovered was that when we produce delta C mutants, so from now on I will refer to this mutant an sp2 delta C, so an sp2 octameric and sp2 lacking the last 17 amino acids, it produced lower cross-correlation function. So in this case, in this assay, I'm showing cross-correlation function 2 interacting RNAs, segments 11, segment 5 in this case, fluorescent labeled. Upon incubation with full length on sp2, we get cross-correlation function equal 1. If we do delta C mutants, now we significantly reduce in cross-correlation function. So approximately 50% of all RNA molecules are no longer interacting under these conditions. Okay, so basically we impaired the RNA matchmaking activity of an sp2 by removing this region. What happens if we look at each separate step of this RNA chaperone activity? Well, we can use additional assay, which is based on a single molecule threat. And now this might be a little bit heavy on single molecule fluorescence. I appreciate that, but these techniques are very crucial for understanding how this RNA chaperone works. And so essentially in this assay, we're using something different. Now we're looking not at intermolecular interactions. Now we're looking at intramolecular, this sort of intramolecular base pairing in this RNA stem loop. And it has a donor in green and acceptor, threat acceptor in red, in orange color. And so if we're looking at this RNA alone, you can see high threat efficiency, as you'd expect, given the distance between a donor and acceptor without any RNA chaperone. Now, if we add an sp2, what you expect is to see lower threat efficiency. So now we started melting this stem loop. And this basically happens at very low concentrations. So about 5 nanomolar in sp2, full length in sp2 is sufficient to start melting the stem loop. Now, if we add delta C, what happens is that a large population of this stem loop now is completely in low threat state, suggesting that actually delta C is more efficient at unwinding this RNA stem loop, which is counterintuitive given what we've seen before. So in essence, delta C seems to be worse at matchmaking, but it seems to be a better protein for unwinding the stem loop. So what's happening here? in collaboration with Dr. Anton at University of Leeds, we conducted several structural studies on NSP2 and delta C. And what we discovered is that we can see which regions of NSP2 are directly interacting with RNA, forming contacts, and which regions are completely lacking any context with RNA, essentially. And so I'm just showing this C-terminal region of NSP2, which we removed, and this unstructured linker region. And what was really surprising is by using hydrogen deuterium exchange or direct RNA protein cross-linking, what we discovered is that in either case, we don't see any direct contacts formed between the C-terminal region and the RNA. So essentially, C-terminal region does not interact with RNA directly. And so Jack Bravo, former PhD student at the University of Leeds, he determined cryo-EM structure of an NSP2, and indeed we can see RNA density. We can't really resolve RNA that well in this cryo-EM structure, but we could see this density that was ascribed to the RNA. And what was interesting is that the C-terminal region is sort of poised towards the RNA, so it doesn't form direct contacts with RNA, but it's facing the RNA. And there are obviously quite a few positively charged residues, arginines, lysines, which are essential for RNA binding, and that sort of explains high affinity binding of unstructured RNA in this case. What was also interesting is that C-terminal region can accelerate RNA dissociation. So in this case, we're looking at dissociation of RNA from either full-length NSP2 or delta C. And what was really remarkable is that, obviously, bear in mind, this is done with relatively short RNA substrates, and we're talking about multivalent RNA binding protein. But even with this difference, which is about 3 to 4-fold difference in dissociation kinetics, we do see dissociation from the full-length NSP2 and hardly any dissociation with a delta C mutant. So essentially, when we looked at the structure more closely, we realized that C-terminal region, in this case, it's poised towards the RNA density, and it also presents several negatively charged residues, in this case, highly conserved aspartic and glutamic acid residues, which promote RNA dissociation from this complex. And so the model we came up with is essentially NSP2 full length is a beta RNA chaperone, because it can both unwind and promote RNA-RNA annealing matchmaking. Whereas for delta C, it's essentially a worse RNA chaperone, because this formed RNA protein complex ends up kinetically trapped and does not proceed all the way to the formation of this duplex, RNA duplex. And because the title of my talk was choreography, molecular choreography of RNAs, just a couple of examples. This is RNA-RNA annealing, and then RNA chaperone, which does not promote RNA-RNA annealing. So now, something slightly different. So here's an sp2, and here's an sp5, which is yet another protein. So the virus, this particular virus, during replication, it doesn't just accumulate an sp2 in the cell, and it doesn't just bind RNAs. It actually forms something what's called viral replication factories. And for many decades, viral replication factories in rotavirus field particularly were called viroplasms. And so essentially the model of this viroplasm is it's a goo of RNAs, RNA chaperone and sp2, and another protein which is intrinsically disordered protein called non-structural protein 5, NSP5. If we do some structural work, if we do thin sections of virus-infected cells within several hours post-infection, we see that these structures are essentially electron-dense structures, and you can sort of see how virus particles almost budding off the surface of this body. And more recently, quite recently, we discovered that if we do live fluorescence microscopy, these entities actually represent liquid droplets containing both an sp5 and sp2, and using this compound, which is commonly used for interrogating liquid-liquid phase separation, we can disrupt this phase, and then you can see the effect immediately. Once we remove this compound, 1,6-hexanediol, we can actually see their reformation. So it's a bit like a shaken lava lamp in cells. I could watch this video over and over again. So what happens actually with the RNA chaperone and RNAs in this liquid droplets? Well, what was really remarkable about these structures is that if we look early infection, so about 4 hours post-infection or later on during infection, when we carry out single molecule FISH, we see GAPDH, highly abundant GAPDH transcripts here in red, and then rotavirus-specific transcripts in green. And if we do analysis of these structures, which we believe are bona fide viroplasms, they're actually highly selective towards viral RNA. So they, despite high abundance of GAPDH transcripts, we do see almost the exclusion of these transcripts from viroplasms, which is not particularly surprising given that virus wants to keep its own cognate RNAs within viral replication factories. And so we can also do slightly different experiment where we use in our cell lines, and these experiments were done by my graduate students, Lilley and Anton, where we looked at localization of either viral RNA within these structures, viroplasms, or we can also do single molecule FISH against all polyadenylated RNAs, and we see pretty much complete exclusion again. So we see polyadenylated RNAs and viral RNAs separated from each other. So now we wanted to look at all RNAs together, viral RNAs, and the way we do this, we employ technique which we call UDEX FISH, but I'm sure some of you must have come across similar techniques similar to MERFISH, where we can do multiplexed RNA imaging in singly infected cells. And the way this approach works is we use essentially single molecule FISH probes against specific targets. In our case, we're interested in all 11 transcripts. They bear small extensions, which we call docking sequence, and they can hybridize to either Cy5 or Cy3 labeled or different dye labeled probes. And by imaging in multiple rounds using spectrally similar or distinct dyes, and by removing these secondary probes, we can re-image and then reconstruct all 11 RNAs in the same locus in the cell. And by doing so, by repeating aligning images, we can do multiplex reconstruction of all 11 RNA segments or more if we wish so. And so just quickly show you some of the results. What this technique allows us to do is that obviously we can look at multiple cells. We started with just all 11 segments together. We can zoom in separate regions and find viroplasms. We can analyze separate viroplasms. And then we can do breakdowns of between cytoplasmic versus viroplasmic transcriptome. It was immediately apparent from this, despite this virus particle producing stoichiometric amount of RNA, we do see non-stoichiometric accumulation of all 11 distinct RNA transcripts in the cytoplasm, and the most abundant being segment 7, segment 6, for example. And surprisingly, in the viroplasmic fraction, we see quite the opposite. We see that segment 1, which is the largest, the longest AU-rich segment, is the most enriched, as you would expect for AU-rich large RNAs that tend to accumulate better in liquid phase of multiple RNA-rich condensates. Although you would probably also imagine that or notice that in this case, the accumulation of all 11 RNAs does differ from cytoplasmic fraction of all RNAs. And so we wanted to understand not just the selectivity of RNA, but also the principles of accumulation or enrichment of these viral RNAs in viroplasms. For that, we started reconstituting viral condensates using recombinantly expressed proteins. And again, this work was done with Lilley. And last year, we published a paper where we essentially characterized protein-rich condensates that are remarkably similar to viroplasms in cells. And they can be formed by mixing NSP5 and NSP2, stoichiometric ratio, and they form these beautiful droplets, which are again sensitive to 1,6-hexanediol. Now, what we're doing, we can also start adding labeled RNAs into this condensate, so trying to sort of move towards real viroplasms or as close as possible. By adding more and more components into these viral condensates, we can start interrogating their RNA enrichment properties, RNA partitioning properties. And what we've seen in vitro is that actually both segment 11 and segment 1 can equally be enriched in this condensate, although we do see the picture is slightly different here because segment 1 as we increase in RNA concentration, it starts dissolving these condensates, which is a typical behavior for RNA-rich condensates. As we increase in RNA concentration, we start observing this so-called reentrant behavior. And so to address this question, why is RNA selectively enriched in sp5 and sp2 condensates, we started doing slightly different experiments where we can remove specific untranslated regions in segments. In this case, we can either produce segment 1 with intact 3' UTR and carry out single molecule fish, and we see that when we preserve 3' untranslated region, which is conserved amongst all segments, we do see nice colocalization of this RNA in this case. However, if we remove 3' UTR and then we carry out single molecule fish, we do see a lot of viroplasmic condensates no longer co-localized with RNA. We can obviously do this with the virus using recently developed reverse genetic system. We can generate recombinant rotaviruses. In this case, we have recombinant rotaviruses bearing EGFP open reading frame, and using this specific virus, we can again discern localization of transcripts containing EGFP just fused to this sequence, poly A-tailed, or we can have this in the context of viral RNA with the intact 3' UTR. And here's the result. Again, we see in this case pretty much no colocalization for polyadenylated RNA and very nice colocalization in case of using intact virus. And I guess I don't think I have that much time, so I'm going to have to go straight to the acknowledgement slides. And I guess thank you for your attention, and I'm happy to take your questions.
Amazing. Thank you very much, Alex. We just made it in time, but are there any questions you might have? Please add them to the Q&A section. For Alex, this is. Don't be shy. There's a question, Alex, from, oh, I'm going to butcher the name, Niva Kaliskan. That's my attempt. Hi, Alex, great talk. Can the difference in distribution be due to difference in hybridization efficiency of the probes? Is the question, Alex.
Yeah, that's a great question, Niva. So yes, it can be, because obviously we don't know how different accessibility of RNA is within condensate and outside. However, I would say we don't do highly quantitative analysis. We can do more quantitative analysis of viral RNAs using slightly different approach, which is called QPaint, which also uses true single molecule imaging. And we do notice slightly different accessibilities within condensates and outside condensates.
Sounds good. Thank you very much, Alex. I think we are just on time right now. So unless there is one urgent question popping up, no, then I would like to thank you both for giving your time and for everyone for listening. And I'm looking forward to the next round of seminars. And I think there was an advertisement early on in the Slack regarding the next event. And bye to everyone. Great job, guys.